Good morning, everyone. Thank you for taking the time to join me today. My name is Tazima Begum, and I lead the Translation Therapeutics Research Uni. As managers and decision makers, you are the architects of this company's future. My goal today is twofold. First, to provide a foundational understanding of the complex, rigorous science that underpins every molecule we develop, and second, to present a compelling case for further investment in our current pipeline. We are not just asking for capital, we are asking for you to invest in the de risking of our future drug development. The concepts we'll discuss today are the very mechanisms that turn a promising compound into a profitable, life changing medicine. Let's start with the basics. What is a drug or medicine in the context of our work? Simply put, it is a bioactive chemical substance. The key distinction lies in its potential effects on the body. We categorize these effects based on the mechanism of action. Number one, the therapeutic effect. This is the intended benefit. Our molecules are precision tools designed to interact with a specific target, a protein or enzyme, to elicit desired beneficial change. This is often described as the drug acting as a key in a biological lock, the receptor. Number two, the physiological effect. This is an underlying mechanism. For example, a drug doesn't just lower blood pressure, it might work by blocking a specific angiotensin receptor, which in turn relaxes blood vessels, leading to the therapeutic effect. Key pharmacological concepts. When we test a new compound, we measure its effect against several critical benchmarks. The first is a placebo. The placebo is a gold standard for clinical trial control. It helps us confirm that our drug's benefit is due to its molecular activity and not simply the patient's expectation of improvement. This is crucial gaining regulatory approval. Next, we address safety and dosing. The therapeutic window. Think of it as a margin of safety. It's a sweet spot between the minimal effective concentration, the lowest dose that provides benefit, and the minimum toxic concentration, the dose at which harmful side effects begin to emerge. Our goal is preclinical and early phase clinical trials is to define the widest possible therapeutic window to ensure a safe and effective product for the market. Key pharmacological concepts. Tolerance is a challenge in chronic conditions. It is a body's compensatory mechanism. It's a biological compromise. Our current research into the receptor allosteric modulators, which don't block the site but change its shape, is directly addressing this challenge by attempting to maintain receptor sensitivity and minimize the development of tolerance. Finally, side effects. These are the secondary actions of a drug. While we aim for perfect specificity, every drug has the potential to interact with targets other than the primary one, leading to off-target effects. We call this polypharmacology. The severity and frequency of side effects directly impact patient compliance and market success. Therefore, improving selectivity is a primary objective in our molecular design and justifies the complexity of the chemical modifications we pursue. Our drug library is rarely built from scratch. We leverage decades of research by working with chemotypes, families of molecules with a common structural core or scaffold. This is a highly efficient strategy. The classic example is the penicillin family. The fundamental beta-lactam ring is the core similarity. This four-membered ring is the warhead. It is essential for the antibacterial activity as it's what inhibits the bacterial cell wall synthesis enzyme. However, the side chain, the rest of the molecule attached to this core, is where the differences lie. For instance, comparing penicillin G to amoxicillin. By simply adding an amine group to the side chain, we get amoxicillin. This seemingly minor change has a huge impact. It dramatically improves the drug's ability to survive stomach acid, allowing for oral dosing, and also broadens the range of bacteria it can treat. This illustrates how small rational chemical changes can unlock significant therapeutic advantages and market potential. Functional groups are the molecular handles we use to fine-tune a compound. These are the keys to entire discipline of the medicinal chemistry, we strategically modified these groups to improve three core areas. Effectiveness. Adding a group like a fluorine atom can subtly change the electron density around the active site, making the drug bind hundreds of times tighter to the target protein. This means a smaller, more effective dose is required. Delivery. To reach an intracellular target, a drug needs the right balance of lipophilicity, fat solubility, and hydrophophicity, water solubility. If it's too water soluble, it won't cross the cell wall membrane. If it's too fat soluble, it'll get stuck in the membrane. We use FGs to tune this balance, improving oral bioavailability and half-life. Number 3, safety. Certain FGs are prone to forming toxic byproducts when metabolized. Replacing a problematic functional group with a biostere, a different group with familiar chemical properties, we can eliminate toxic pathway, dramatically improving the drug's safety profile without sacrificing efficacy. Moving from 2D structures to the reality of 3D space, we encounter isomerism. This is a fundamental to a drug's performance as the body's receptors are highly selective 3D structures. Geometrical isomers, or CIS, and transisomers are molecules whose atoms are arranged differently in space around a double bond or coordination center. The difference is profound. Consider the inorganic compound cisplatin, a highly effective chemotherapy agent. In cisplatin, the critical ammonia and chloride ligands are arranged on the same side. This specific orientation allows the molecule to bind to two adjacent sides on the DNA strand, causing the structural kink that triggers cancer cell death. The transisomer transplatin, where the ligands are on opposite sides, is completely inactive as a cancer drug. It cannot interact with DNA in the necessary cytotoxic way. This demonstrates that geometry is function. The principle is a daily consideration in our work. Manufacturing and isolation processes must ensure that we purify correct active geometrical isomer. If geometrical isomerism is about front and back, optical isomerism or chirality is about left-handed versus right-handed molecules. The analogy is simple. Your left hand and right hand are mirror images, but you cannot perfectly superimpose them. Our biological receptors are also chiral. They are likely a right-handed glove. A drug's therapeutic effect requires a precise fit into the chiral receptor's pocket. The molecular handshake, often only one enantiomer fits correctly. Consider ibuprofen. We administer it as a racemic mixture. However, the S enantiomer is primarily responsible for the anti-inflammatory effect. The R enantiomer is largely inactive, although the body does slowly convert a small amount of R to the S form. Once a molecule is synthesized and tested, we need to detect, quantify, and measure its purity. This requires highly sophisticated analytical techniques. Our primary workhouse is HPLC. It is a gold standard for separation and quantification. It tells us how much of our drug is presented in the sample, e.g. plasma, and crucially, it verifies that our compound is pure and separated from any inactive starting materials or byproducts. But we don't need just quality, we need absolute identity. This is where mass spectrometry comes in. The MS in a molecule fingerprinting tool, it accurately measures the exact mass of our molecule and its fragments. When we coupled with HPLC MS, we achieve unparalleled sensitivity, allowing us to detect and identify minute quantities of drug and its metabolites in a complex biological matrices like blood and urine, providing the rigorous data required for regulatory submission. identifying new drug candidates. The question is, how do we find the initial lead molecule? We employ two primary strategies. Number one, HTS. This is a brute force industrialized approach. Using advanced robotics, we can screen our entire compound library against a disease target looking for a hit. Any molecule that shows initial activity, this technology is capital intensive but dramatically cuts the early discovery timeline. A more precise method is rational drug design, where we use structural knowledge. Number two, target based design. We leverage protein structure data to design a perfect fit molecule. This reduces the reliance on random chances and allows us to engineer specificity from day one. The initial investment in structural biology is high, but the payoff is a cleaner, more tailored lead compound with a higher success rate. We also maintain a strong program in natural products screening. Number three, natural products. Nature is the most prolific chemist. We screen natural extracts because evolution has already optimized these compounds for biological activity, often yielding novel scaffolds that synthetic chemistry has not yet conceived. This brings me to the critical role of computation in silico design. This is where our current funding proposal is the most impactful. We use powerful algorithms for molecular docking. The computer simulates how millions of molecules will physically fit and bind energetically into the target receptor's pocket. This process allows us to virtually screen compound libraries that are impossible to screen physically, instantly prioritizing the most promising candidates, thereby saving immense time and money on synthesis and physical HTS. Beyond simple screening, we utilize de novo design. This is generative AI for chemistry. The computer design brands new compounds perfectly tailored to the geometric and electronic properties of the active site. Is truly rational design, enabling us to leapfrog traditional trial and error synthesis. The interrogation of our physical lab work with cutting-edge computational tools is a competitive advantage of our unit, but it requires continuous investment in hardware, software, licenses, and specialized personnel. In summary, drug discovery is an exacting science. Every successful molecule is the product of meticulous chemical engineering, rigorous analysis, and an acute understanding of the body's response. We understand the importance of shear purity and geometrical orientation because the wrong isomer can be inactive or dangerous. We rely on HPLC-MS for absolute proof of our work. Our process is now focused on efficiency using HTS to find initial activity and molecular docking to engineer support compounds. By investing into a high priority phase 1 initiatives, you are not simply funding an experiment, you are doubling down on efficiency, safety, and market readiness. The capital directly enables us to apply the advanced analytical and computational methods discussed today, accelerating our lead candidates past the riskiest stages of development and bringing us close to a higher value, regulatory ready product. The chemistry is sound. Your investment is a catalyst to turning science into success. Thank you. I am happy to take your questions. So here is my references list.